Hello and welcome to our podcast Pager. I'm George Milner.
And I'm Stan Dale.
Tune in to hear us talk with some amazing people about the subjects changing medicine today.
From stem cells to mapping the brain and medical ethics, we hope to give you a glimpse of the bright future ahead through discussions with some of the people making it happen. Hey everyone, welcome back to another episode of the podcast. This week's guest is Neel Aluru. Neel's an associate scientist at the Woods Hole Oceanographic Institute, which is a world-class ocean research institute on Cape Cod here in Massachusetts. And Neel leads a group that looks at how chemicals in the environment, and in particular those from the ocean, affect human and animal health. In particular, how epigenetics can be changed by toxicants in the environment during development, and what the effects can be on adult health. This is a concept referred to as the developmental origin of health and disease. So in this podcast, Neel and I explore what this concept of adult disease being rooted in development means, how scientists investigate chemicals in the ocean, a particular class of chemicals called polychlorinated biphenyls, or PCBs, and their effects on genes in the brain, model systems Neel's group uses, gene editing tools and the future direction of this field, and some interesting case studies from history. So without further ado, I hope you enjoy our episode with Neel Aluru. Neel, thank you so much for coming on the podcast and giving up some of your Saturday morning.
Yeah, thank you for contacting me. I didn't know anything about you before this.
Perhaps a good place to start would be, if you could just fill us in on sort of your background and how you became interested in research into oceans and how it might affect human health. I know your research kind of extends beyond that, but just to give us a background.
Yeah, my background is mainly, my undergraduation was in fisheries science, trying to learn all about aquaculture, fisheries management, to fish processing technologies. So I was mainly interested in following the field of agriculture and veterinary science when I started my career. And then after I finished my undergrad, I got a master's in international fisheries management at University of Tromsø in Norway.
So it really was all about the oceans from the get-go.
Yes, definitely to get go. Yeah, it's mostly undergrad was mainly focused on aquaculture, learning the basics of, basic biochemistry of fish and fish physiology. And then when I was in Norway, even though my coursework was all on fisheries management, I ended up working on field of toxicology, how environmental pollution affects fish species. I was working on a very interesting species of Arctic char. So it's a salmonid fish, but its lifestyle is anadromous. What that means is these fish spend almost 10 months in a year in fresh waters where they are landlocked, completely frozen. And for 2 months, they go to ocean where they voraciously feed, gain double their body weight, come back, and then they don't eat for almost 10 months.
I mean, that's fascinating. They must be very differently adapted to surviving in those two environments.
Exactly. So they they store all the fat, and then they survive on that. So the interesting part is a lot of pollutants, which are lipophilic. They bind to these fats and then they stay. So when you're consuming the fat, when you're breaking down that fat, these pollutants starts moving around in the body. So you store your fat in the adipose tissue, and then when that is being utilized, the pollutant starts finding the spots where there is fat. And one such area in our bodies, including all the animals, is the brain. Brain consistently has some fat deposits. So these pollutants, the lipophilic ones, they mobilized and then they go to the brain. And obviously, when you have pollutants in the brain, you can imagine the effects on all sorts of behaviors, not only reproduction, just neurobehavior, the motor function, to when you think of wild animals, you can think of escape reflexes, predator avoidance, etc.
I was going to say, you can see how very quickly the chemicals in the ocean and the health of sea life is very inextricably linked to human health. Because
exactly, precisely.
If anything, marine life almost acts as a mechanism for concentrating some of these dangerous chemicals, which we then consume in the fish.
Yeah, precisely. Yeah, that's how there are a lot of good examples of mercury contamination to uh, for pregnant women are not allowed to eat too much fish.
Too much wild fish.
Yeah,
Hmm.
not just for
And so
mercury purposes, but there are a lot of other contaminants now.
it's interesting that your sort of field of research was relevant to this, right from your masters. I suppose my one question would be the phrase aquaculture. I haven't heard before. I work with cells, so I'm sort of imagining that it's the equivalent of cell culture. It's keeping fish in
Exactly.
tanks.
It's precisely because as one of the cheapest forms source of proteins is seafood, and it's the most nutritious. Lot of omega-3 fatty acids or unsaturated fatty acids, uh, especially when you want to feed 6.8 billion people. Uh, when you think of developing countries, where
Mm,
just for India there is a population of 1.3 billion, you can't feed just based on fishing. So you need a source of protein that is cultured. So aquaculture is a huge thing, not only in the developing countries but also in developed world. So if you go to a market, most likely you're going to find farmed salmon, for example. So it's very expensive if you want to buy wild
well,
salmon.
and you can see how immediately the relationship between the consumption of fish and something like the effects on the brain become complicated because as it's sort of quite well known, omega 3s, unsaturated polyunsaturated fats are known to be good for kind of the health of neurons in the brain. So I'm sure this quickly gets a bit more complicated than it seems at face value.
Yeah, and because these mechanisms are highly conserved. Same thing when you consume or when you are exposed to humans, when humans are exposed to these pollutants, they generally end up in fatty deposits, and the effects on the brain are so immense that there is a huge story, at least in the United States, in the 50s and 60s. They have used to seal the windows. They used a caulking material that's made of PCBs.
Mm.
So the exposure to these caulking materials in elementary schools and their effects on children, their development, their brain development, they're all well understood now.
Mm. That's quite a useful case study for understanding how these PCBs, polychlorinated somethings,
Biphenyls, yeah, yeah, biphenyls, polychlorinated
biphenyl,
biphenyls, yeah,
are affecting human health. But
yes,
I
exactly.
suppose before we jump ahead of ourselves, so walk us through how you end up at Woods Hole and doing the
Yeah,
research you're
so I
currently
did
doing.
my PhD in Canada at University of Waterloo, where I went more in depth on toxicology, trying to understand how chemicals affect our stress response. So we
Mm,
have this flight or flight response. How is chemical exposure can affect the endogenous stress response behavior all animals share, including humans.
So
So
now you've gone from thinking about toxicology in fish,
yes,
about what might be the effects on human sort of sympathetic stimulation and the activation of our sort of fight or flight systems.
yes,
And interestingly, you've sort of got to look at the same body of water from both ends, both the European and the North American side of the Atlantic, which I think is quite interesting.
yes. But most of my PhD was using a species called rainbow trout. Again, it's a salmonid, but my main work was all experimental, where we had tanks of fish, where we can manipulate and conduct experiments and understand the process. I did not do much of field work when I was doing my PhD. And as I was
Okay, I
doing my
see.
PhD is when I got into this experiments where when you expose females, or for example mothers, to pollutions, and then you look at the offspring, what are the effects? Is there a maternal transfer of the contaminant dependent adverse effects?
Mm. And
So
I think Neil, here we've struck upon kind of the crux of what this conversation is going to be about.
Exactly.
The developmental origin of disease and the relationship that chemicals in marine life and in the oceans have to it.
Yes.
Um, and so at this point, perhaps it would be useful just to explain what we mean by the developmental origin of disease.
Yeah. So
It's
what it's a paradigm that's been being studied for the past 20 years or so. It's called developmental origins of adult health and disease. So one of the nice examples is, for example, smoking or drinking by pregnant women.
mm,
How the effects can be, it can cross the placenta and affect the offspring. So what they found recently is that some of these effects cannot won't be acute, so you don't see the effects in the offspring soon after birth. They develop normally, everything is normal, but once the offspring becomes an adult later on in life, then you get to see a lot of neurobehavioral deficits, metabolic deficits. So and then they started to link how the you can reprogram how development is such a sensitive period
mm.
during which your programming is happening, your cellular differentiation is happening, tissue differentiation is happening. So during that time, any disturbance, any kind of exposure to stressor can have reprogramming. Again, the word reprogramming is very hard to define in terms of whether it is genetic reprogramming or epigenetic reprogramming. There are many different ways.
Mm,
However, you are disturbing the system so that the effects are subtle, but they are long term, they are
mm.
persistent for a long period.
It's quite remarkable to think about, really, and almost quite insidious if you really think about it. You can see how it took a long time for people to have an idea that this might be having an effect, small effects during development, whilst the embryo is developing, whilst the fetus is developing, postnatally, can have such profound influences on later life.
Yeah, exactly. Yeah. And not only that, these studies were initially focused on the mothers, but now we know, right around conception, if the sperm is exposed to chemicals or exposed to contaminants or any kind of stressor, that also can affect. It's not just maternal effects, but you can also have these long term effects can be seen due to deficits or disturbances in male reproductive system.
The sort of the butterfly effect analogy seems appropriate in sort of a really perhaps depressing sense. Interestingly, I think a lot of these studies back in the day were really more epidemiological in nature. It's really interesting to see how this field is now progressing to understanding what's happening at a cellular or intracellular level, and how that's probably going to give us a completely new level of resolution in
Precisely.
understanding the mechanisms.
Yeah, no, definitely. Most of it is based on epi work.
Mm,
There are amazing number of studies now showing correlations. Even if you have one cigarette smoke at certain times, you can alter a signature in your DNA that is so persistent. So people are using that as a
mm.
marker in order to study the effects of smoking.
Smoking might be quite a good comparison for then thinking about the chemicals you're thinking about, because I suppose, particularly as someone who thinks about medical science and
Mhm.
epidemiology, typical epidemiology that people study, you think about kind of more direct and obvious environmental influences. And kind of my understanding of this literature is, you're thinking about oxygenation to the fetus, nutrients that the mother might be taking in, and so there's a whole range of environmental influences which we don't really pay that much attention to, but which might be having quite profound effects, and to kind of the ocean and the effects of marine life.
Yeah, no, the it's amazing the number of stresses that have come to focus these days. You're right. Hypoxia was, one of the important things. After there are a lot more now, various chemicals. For example, PTSD,
Mm,
post-traumatic stress disorder, And one of the interesting studies is about licking and grooming. I'm not sure whether you are aware of these studies where people have done experiments in rodents where you take the pups from a mother, and then you can see if the mother is licking and grooming the pups, they have lot less stress when they become adults compared to the pups that are not given that much of parental care. And we know that those effects are literally focused on one gene. The mechanism is based on one gene being epigenetically modified in the brain, in a specific region of the brain.
That is remarkable to think about such a macro behavior,
Precisely.
and translating to such a specific gene. That
Yeah.
really is amazing.
Yeah.
I'm now thinking about where best to kind of take this conversation, because there are about 5 different directions I feel like we could pull it. The first thing I think I'm going to ask you about is like, okay, there's an ocean full of chemicals which might be affecting human health.
Yeah.
How on earth do we go about screening for things which might be especially important? What is that process like? Is that something you're interested in?
Yes. Again, identifying chemicals is pretty hard. There
Mm,
are a variety of chemicals. Uh, and then again, this is one area where it's $1 million question in the field, that is the study of mixtures. We are never exposed to single contaminants in our
mm,
entire life. For example, just think of us. I'm sitting on a couch, which I'm sure has flame retardants on them.
mm,
And then there are other things that are around me in the air that I am breathing now.
mm,
So identifying chemicals is an entirely different question. It's an important question to understand what are the various individual what's the chemical composition of the water near you, for example.
mm.
um,
And
and then
how X might summate kind of non-linearly if they're acting on downstream pathways which are conserved.
precisely, that's a question. So I think the approach I am taking and many others at the institute are taking is trying to understand the what is the mode of action of individual group of chemicals.
Mm,
My main focus so far has been on polychlorinated biphenyls. These are legacy. They are banned. There is no production currently anywhere in the world. Definitely in the developing world. However, they are legacy contaminants. They don't degrade. They have a really long half life, so they are persistent still in the environment, and they'll be there for a long time.
so we're really living with the effects of sort of bad decision making done decades ago, presumably.
Yeah, yeah. So we are trying to understand, again, within PCBs you can divide them into two different groups. The ones that the chemical structure looks more like dioxin. Like dioxin is the chemical that's used in Vietnam War, Agent
Mm,
Orange, if you're aware of it.
it's what they sprayed the kind of the foliage with
Yes,
to.
precisely.
Mm,
So those group of chemicals, dioxin like PCBs, they do behave very differently, and there are almost 50 to 60 different congeners, and they all behave the same way. So we are trying to understand what is the mechanism of action, and we know they persist in the environment and they are there in our blood as we speak. So there are studies by Haynes, one of the NIH institute, where they have collected blood from humans, and we know that there are relatively low levels. However, they are present in almost 95% of the population.
mm. Just to
Uh,
give us an idea, when you're thinking about some of these chemicals, how low can the concentrations get per deciliter? I imagine, in some sense, this area of research is made more difficult because you're looking for needles in a haystack, and then trying to think about the effects that might be having.
yes. So when you think of any of the toxicology studies in any model system for that matter, uh, 10 years ago, most of the focus was not on environmentally realistic concentrations. People like me mainly focused in understanding the mechanisms,
Mm,
and we always used pretty high concentrations in order to see a clear phenotype.
mm.
either a behavior or a malformation, so on and so forth. But now, in the past 10 years, the whole field has shifted. We are literally looking at environmentally realistic concentrations. Lot of studies, and then in toxicology there are many different types of responses. If you use a very high dose, you get a one effect. If you use extremely low dose, you get a very different effect. So depending the dose makes the poison,
Mm.
they say.
That's, I mean, it's fascinating, and I think it shows how difficult that kind of research, or the complexity of that kind of research. It makes me wonder, sort of one question that could be asked is, do some of these chemicals acting via epigenetic pathways have kind of continuous or discrete functions as you continue to increase the concentration? Like, is it that the tiniest concentration will lead to the tiniest decrease in the health of an area of the brain? Or is it that actually you need to reach a certain threshold for cells to be disturbed and their epigenetic marks to be changed?
Yeah.
It's sort of a difficult question to answer, I imagine, but
Yeah,
maybe some
so I
people
think you're bringing a very good point here. So l- let's not, even if we talk about phenotypic effects, looking at the whole organism,
Mm,
we still don't know clearly what these thresholds are. And every chemical compound behaves little differently. And when you think of epigenetic effect, the whole field of epigenetics, especially in understanding the effects of environmental stresses, is still relatively new.
mm,
When you think of chemical effects, people are still trying to understand what are the effects, and we really don't. There are very very few studies where people have done multiple different concentrations and then trying to measure, is there a linearity in any of these effects, or is there a threshold? So
mm.
yes or no answer to your question is we really don't know.
Yeah. Yeah, I think I agree with you. The brain is the thing that we most desperately need to protect. Um, Before we go on to thinking about, I want to ask you about kind of the model systems you use. Uh, you mentioned Agent Orange, and we spoke a bit about epidemiological studies. Are there any really other useful sort of case studies which nicely show the effects of specific toxicants on our biology?
Yeah. So another very good example from wildlife is exposure to DDT, which
Mm,
is a herbicide which was used, again, that's now banned now. There are a number of classical examples. For example, now I'm trying to think, imposex in snails, that is
mm,
due to exposure to tributyl tin. The boats have a coating in order to anti-fouling coating that's there
mm,
on the outside of the boats, and it used to have a lot of tributyl tin. And when these snails attach to them, they are exposed to this compound, and it affects the development of the sexual organs. That's
mm.
one example.
So it's
Uh,
not just the sea poisoning us; we're poisoning it back.
Oh yeah, yeah. I don't think there is any place which is more pristine these days.
Mm,
Another example is a lot of uh, trout and salmon in Great Lakes because of dioxin, like Agent Orange compound. They have cardiovascular defects. That's a classical example
mm,
where people have studied, oh, why are these fish dying, why are the larvae not developing? You can see their
mm.
edema around the heart is so bad when
Yeah.
exposed to contaminants.
And perhaps even bad sort of toxic algae blooms on freshwater surfaces.
Yes, microcystins, cyanobacterial toxins. So that's another area of focus here at the institute. Is there is a huge group, very well known group focused on harmful algal bloom toxins.
Mm,
We are just getting into it, trying to understand the the toxic effects related to human health.
mm,
But people here have been measuring the species that produce these toxins, and how much is produced during what times of the year, and what physical processes in the ocean, how do they affect the production of these toxins.
Mm. Now, let's think a bit about model systems. Say we want to study any of these chemicals, we have to do it in some sort of model system. And to sort of set the stage,
Yeah.
this really is a very difficult set of questions to answer. You're trying to map environmental exposures to developmental changes or changes of activities of genes in cells to
Yeah.
adult phenotypes later in life. I mean, I don't think off the top of my head I can think of anything which would be trickier to sort of show a causal relationship between.
Yeah.
What are some of the models we have or you have at your disposal, and sort of what might be some of the pros and cons of each?
Yeah. So regarding model systems, obviously, again, this question brings another point here is most of the funding agencies support research related to human health. That
Mm,
is the ultimate goal. If you say that you want to study on environmental health, there are not many uh, avenues for getting funding.
mm,
Yeah, that's the way life works here. So
mm,
people have used rodent models for a long time because they are, even though it's not 100% true, but mainly people focused on rodent models because they're mammals and da 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 da, and you can translate to human easily. But yeah, but we can argue that that's not the case.
mm,
And the reason why we focused on zebrafish for some of these studies is because zebrafish are excellent because they have a very short generation time.
mm.
From embryo to adult, it only takes 3 to 4 months, and they produce tons of embryos or eggs because each pair can give you 100 to 200 eggs easily every week.
And eggs are presumably better than anything happening in like sort of any sort of development in vivo, so to speak,
Yeah,
because you can
exactly.
probably more easily manipulate and take a look at what's going on.
Precisely. In case of rodents, it's in utero, whereas in zebrafish, the fertilization happens externally. Eggs are released and sperm is released into water, and the fertilization happens. So we can actually collect eggs when they are in one cell stage.
Mm.
They're transparent, so you can actually observe one cell divisions, and it's amazing. You can Google it to find the developmental. There are very good time lapse images showing the whole development.
And lineage tracing must be quite easy with fluorescent reporters and
Yes,
things of that nature.
precisely. The transgenics, we are using transgenic these days in order to study microglia, the non-neuronal cells in the brain. We have lines. You can actually study myelination in during development.
Wow.
So there are numerous advantages of using zebrafish as a model in order to study. And the question about translation to humans, the developmental processes are same. The Obviously the organs doesn't look exactly like in humans, and there are a few other small differences. However, almost 70% of the protein coding genes are conserved across all vertebrates.
Mm.
So definitely there are advantages. The way I look at it is, as long unless you are studying in humans, all the other model systems are complementary to the human systems. Even in
Yes,
terms of
and I
cell
imagine they sort of carry different strengths and weaknesses. You know,
exactly.
talking
So
about zebrafish and
yeah,
and mouse models, zebrafish are obviously much higher throughput, and like you said, it's a shorter period of time
yeah,
between
exactly.
developing the adult. So, what I imagine is that in a perfect world,
Yeah,
you'd be able to do earlier studies looking at large ranges and combinations of chemicals in something like a zebrafish,
yeah,
and then ultimately you'd sort of refine those findings by using mouse models or primate models.
yeah. Definitely, you can't base your results just using one model system. You
Mm.
need to have multiple models.
Mm.
Yeah,
Thinking now again about zebrafish,
yeah.
PCBs, and the brain,
Hmm.
what might the effects of PCBs be on the brain? Which functions are impaired? Do you see any changes in the structure of the brain?
There are several studies. Like definitely, we know quite a bit about how PCBs affect neuronal development.
Mm.
Uh, and to some extent, we also know about the non-neuronal cells in the brain, the glial
The
cells.
glial
Yeah,
cells. Mm.
yeah, yes. Again, there are a number of studies showing how it affects the production of how PCBs affects the pituitary gland or the
Mm.
hypothalamus.
Which, just to refresh people who might not be aware, these are pathways involved in producing peripheral hormones.
Yes, so yeah, these are the regions in the brain that are very critical in the production of hormones
Mm.
that stimulate various endocrine glands like testes, ovary, adrenal gland,
Mm. So
thyroid gland.
your research on PCBs in the brain has looked at changes in behavior in zebrafish, or what specifically have
Yeah,
you been
so
looking
a few years ago, we conducted a study where we looked at how exposure to very, very low levels of PCBs affects uh, affects behavior later on in life.
at? Mm.
So we exposed the zebrafish embryos for a very short period to very low levels of PCB, and then we raised them for 6 months later as adults. When you look at their behavioral responses, you can see drastic differences among them. And then we tried to correlate those changes to see, okay, if that is the case, probably we should measure the genes in the brain. So
Mm.
when you look at the gene expression changes, most of the changes are metabolic effects.
Interesting.
Yeah.
Which is then affecting the brain. And with some of these effects, is it that you're shifting the behavior? Say you plotted it. Are you shifting behavior, say left or right, in terms of probability of a given behavior, or is it just a change in variability, for example? And does
Uh,
that change between behaviors? Maybe this is a question that's a bit esoteric, but it just popped into my popped
yeah.
into my
No,
mind.
I think no, it's a good question. Again, you can do many different behaviors. Again, the results we found was accidental in a way. So the idea was we were looking at a simple assay where we are trying to assay whether these. So there is one widely used assay called novel tank assay.
Okay.
So this assay literally measures uh, the effect of whether animals how quickly they adapt to the novel environment. So
Okay.
when you take a fish and put them in this novel tank just by itself, initially one of the first behaviors they show is flight or flight response.
Mm.
So they go to the bottom of the tank and then they're, where am I? It's a very novel environment. So
They're
they
just
show
kind of
this flight
hunkering
or flight
down and
response.
waiting to see what's going to happen, I suppose,
Exactly.
at first.
So within
And then
the
presumably
initial
they start
few,
to, let me take a guess, they start to explore a bit more, and sort
precise,
of you can
precise,
see changes in the rates of their exploration and how
yes,
well they learn. Mm.
yeah. So you can actually measure how long these animals take to get out, start exploring, for example.
Mm.
So the exposed fish, initially they had a hard time starting to explore the environment. But however, there were not huge differences between the exposed and unexposed groups. But when you repeat this assay every 2 weeks later, we observed that the PCB exposed fish are not habituated to this novel environment. Whereas the control animals, 2 weeks later, when you repeat this assay, these fish were able to recognize the environment and then try to explore pretty quickly. Yeah. And you can do many different assays. They are pretty complementary to the assays that have been done in rodents. Again, it's challenging to do many different assays all
So
in one study.
yeah, and I suppose you ought to pick the ones that you're kind of the most closely track what you're really interested in. But I imagine it's quite useful that some of the studies are quite transferable between different model systems. You can look at the
Mhm.
same thing in different
Yeah.
stuff. Yeah, fantastic. Two of the things I wanted to talk about as well. We'll get to one of them first, and hopefully the other. The first one is, what questions you might be interested in thinking about specifically using the model systems you're using and the work that's coming up in your lab and what's exciting you. And secondly, thinking about, what tool, we talked about the animals you can look at these effects in. What tools do we have for looking at epigenetic change and DNA methylation?
So, in terms of approaches for measuring epigenetic marks, either it is DNA methylation or chromatin modifications or non-coding RNAs for that matter. Nowadays, I think in the past 10 to 15 years, these studies are possible in any model system you can think of.
Mm.
Genomes are available. For example, for zebrafish genomes, genome is known. Even for killifish, I have some data on DNA methylation. I haven't published it yet, but by bisulfite sequencing, these days you can even do whole genome bisulfite sequencing in any species of interest. You can study chromatin modifications. There are multiple approaches, sequencing based approaches.
Sure, yeah. That sort of, it makes sense that you'd be more likely to pick up an effect if we can look, silencing of whole genes as opposed to single bases. Um, yeah, I mean, it just seems like a whole new world of mechanisms for looking at this stuff. I mean, is CRISPR like a valuable tool in particular? Can you use that to see whether knockout of a gene has the same effect?
Yes. Yeah. Yeah, we have been using. I don't know how much you are aware of CRISPR and gene editing tool history. We initially started using zinc finger nucleases. That's the first iteration, and then we skipped talons because we were struggling to do some of the work using zinc finger nucleases, and then the talons came, and then we literally jumped from ZFNs to CRISPR. So I have generated at least 10 different CRISPR knockouts in zebrafish, and we have 3 different knockouts in killifish. So yeah, these technologies have definitely helped us to study the role of genes, reverse genetic approaches in any model system you can think of.
Mm.
Yeah.
I sort of, it's making me think you almost went from like a camper van to a sports car in terms of ability to
Yeah,
change genome.
precisely. Yeah.
Yeah, and are you able to knock out things in a tissue specific fashion using Cree-Lock stuff? So
That's a good question. No, we haven't done any tissue specific knockouts, but last week I was discussing with my colleagues that we should do tissue specific knockouts
mm,
using locks recombinase.
particularly if I'm thinking about, say, I was interested in a metabolic phenotype, there
Yeah,
are so many
precisely.
ways involved in what that might look like. We mentioned the pituitary, the liver, you name it. So it would be quite interesting to pick out, well, which programming effects on which organs are contributing what to this phenotype.
Yeah, I agree. Yeah, no, I think the tissue specific knockouts is something zebrafish world is just taking up.
Fantastic. Well, I think we've got through all the kind of technical aspects I
Yeah.
was really
Mm.
looking forward to, and thank you for talking about all of that.
Sure, no problem.
Other just sort of randomer questions I'd like to ask you. Like, to me, the Woods Hole Oceanographic Institute sounds like a really cool and exciting place to work. Do you get to go out on any kind of expeditions? It must be a real treat to work with people doing all sorts of stuff with marine life.
Yeah, no, it's a wonderful place. I highly recommend you visiting Woods Hole sometime. Just during summer, it's buzzing with activity.
Mm.
There are a number of institutes around here. It's a
Well,
very
I'm
small.
taking it as an invite. You have to show me around the lab
Yeah,
when.
yeah, yeah, for sure. Uh, yeah, when everything gets back to normal, for sure.
Mm.
Uh, yeah, it's a good place to work, and there are around 1,000 people working with a lot of engineers, and it's a diverse fields of expertise that's available. There are mathematicians, there are modelers,
Mm.
there are biological oceanographers who literally go to the ocean. That's their expertise. Unfortunately, most of my work is lab based, and to some extent field collection, collecting killifish, for example.
So I'm picturing sort of the lab and marshes around Massachusetts rather than the open Atlantic.
Yeah, yeah, yeah.
Yeah.
Most of it is that way.
What are you particularly excited about with respect to the future?
I think I'm mostly trying to focus on DNA and RNA modifications.
Mm.
I'm not sure you are aware of. There is whole field of RNA modifications. The modifications in RNA can affect the stability of the transcripts or the turnover of the transcript. So we have known there are almost more than 150 different RNA modifications, and lately there is some focus in understanding the role of these modifications on various neurobehavioral deficits. So I'm slowly getting into this field of RNA modifications and trying to see how we can use zebrafish as a model to study some of the effects.
That's fascinating. So the developmental origin of disease, of
Yeah,
health rather,
yeah,
might not just be taking place at the genetic level. We might also have to think about RNA.
yeah, precisely. So epitranscriptomics is slowly getting some attention. As I said, there are like 150 different modifications. I'm now focusing on adenine 6 M6A modification, methylation of adenine at the sixth position. That seems to have, there are several studies showing the modification of this particular modification having effects on circadian behavior, for example.
Mm. I imagine it might play a role in the initiation of translation being that far.
Yeah, yeah, precisely.
Yeah.
Mhm. So the measuring them is little tricky. uh, we just published a paper measuring this particular modification in response to exposure.
Mm.
When you expose uh, zebrafish embryos to PCBs, uh, this modification is altered in several genes associated with aryl hydrocarbon receptor signaling.
So it all comes full circle.
Yeah, yeah, yeah.
And
So that is the beauty of the system. If you want to understand the mechanism, you definitely need to know the mode of action of the chemical.
mm,
So that makes it easy to understand, and it has, you can relate these results both to human health as well as the environmental health or the,
yeah, no, it
yeah.
sounds interesting stuff. I'm looking forward to reading it.
Hmm.
I think on that note, we're probably running out of time. So let me
Yeah,
just say a huge thank you for coming on the podcast, and I hope to see you in at Woods Hole at some point.
yeah, no, thank you for inviting me. It's a pleasure to talk to you.
Thank you for listening to the Pager podcast. If you've enjoyed it, then head to Spotify or iTunes to hear more.
As ever, we'd love to hear what you think, and you can reach us via our Facebook page or by email. See you next time.